To process a sputum sample for Truenat testing, the cleaning of workbench and instruments should be done first. Disinfectants used are freshly prepared 1% sodium hypochlorite and 70% alcohol. Clean the working surfaces unidirectionally, first with freshly prepared 1% sodium hypochlorite and then with 70% alcohol. Discard the cotton and paper towel used for wiping in the yellow bin. Clean the TruePrep Auto V2 instrument with a paper towel wet dipped in 70% alcohol. Discard the paper towel used for wiping in the yellow bin. Cleaning of the workplace and machine is to be done once a day before starting any work on the workplace. Discard the gloves used while cleaning into the red bin and put on a new pair of gloves. Take out the sample pre-treatment kit containing the reagents stored at room temperature. Arrange the samples in sputum cups or falcon tubes and the pre-treatment kit on the workbench. Unpack the TruePrep Auto MTB sample pre-treatment pack. It contains disposable transfer pipettes, 9 ml liquefaction buffer and 2.5 ml lysis buffer. Open the sputum container cap gently and place the cap on work surface upside down. Volume of sputum can be 2 to 5 ml. Add 2 drops of liquefaction buffer to the sputum sample. During this step, care should be taken not to touch the nozzle of the buffer bottle to the sputum container. Cap the sputum cup and swirl it gently for proper mixing. Keep for 10 minutes at room temperature. During this time, swirl mix the sputum container every 2 minutes for proper liquefaction. If liquefaction is not complete, keep for additional 5 minutes. Take out the lysis buffer bottle, uncap it, pull out the bung and discard it in the red bag. Label the lysis buffer bottle with sample ID. Transfer the liquefied sputum sample into the lysis buffer. For this, take out a sterile 1 ml transfer pipette from the package. Do not insert hands into the package of transfer pipettes as this could contaminate all the pipettes. Collect 0.5 ml of liquefied sputum sample by pressing bulb of the transfer pipette and add it into the lysis buffer bottle. Do not drop the sample into the lysis buffer bottle from a height. Discard the transfer pipette in the red bin. Close the lysis buffer bottle and shake well for mixing of liquefied sample with lysis buffer. After keeping for 5 minutes, the sample pre-treatment is completed. We can now proceed for DNA extraction.